Hi, my name is Matthew Dickerson. I'm a lead scientist at BioScale, working with the company for several years developing this new technology to analyze protein and protein interactions, specifically around cell lysates and kinases and those kind of things. This poster is some development work that we've been doing internally to understand how we can use the platform to really do a large analysis of kinase activity, specifically around oncology. So first I'd like to lead you through what we've been doing. So what we set out to do was to develop a set of assays in which you can really look broadly against multiple kinases and multiple kinase pathways. And so that's what we've illustrated here. And so what we've done is that we've developed assays against the phosphorylation of the MEK kinase, the phosphorylation of ERK, the phosphorylation of P38, phosphorylation of J&K, as well as the phosphorylation of AKT. And that allowed us to look at 4 different pathways simultaneously. So how did we set out to do that? Was that we first would, in order to develop these assays are classic sandwich assays in which you have 2 antibodies coming together to bind to the analyte and detect the analyte in your sample. And that's done in our technology all as one good gemisch. So we put everything into a well at the same time. So we're simultaneously measuring everything in the state in which it's presently presented to us in the sample. So the way that we do that is illustrated here. We start by developing an assay by buying in antibodies from commercial vendors. And then what we do is we do a pair analysis for the assays that will function best in our technology. And so this is an illustration of a set 96 well plate. Now in 96 well plate, of course, you have columns and you have rows. And so what we've done is in the columns, for the illustration purposes here, we're illustrating the assay development for the phosphorylation of P38. Now what we've done in the columns is taken antibodies that are specific for total P38. And then in the rows, we've matched those with antibodies that are specific for the phosphorylation of P38. Now those 2 antibodies should come together when the analyte is present and light up and give us a positive signal. Some antibodies will work better than others. So what we've done here is done a quick screen, and you can see what we've illustrated are these pairs are the ones that work better, the best for our system. Now once we've identified the pairs that work best, then we have to go ahead and develop an assay. And that's what's illustrated down here. Now these are several different assays that we've developed against kinase markers. Let's look first at the phosphorylation of the EGFR receptor. And what we do is standardly people to do this kind of analysis would run a Western blot. So the Western blot is illustrated here. In a Western blot, you load a certain amount of protein, you blot it against your antibody, and see your band. So that's what's here. So we started with 5 micrograms of protein, you dilute it down, and we're trying to compare our sensitivity and this graph to the sensitivity of the Western blot. And so here we've got 5 micrograms of protein, and we're comparing to 5.5 micrograms of protein loaded in our assay. And because we have a lot more sensitivity in our technology, we're able to do that leap. And what we've done is we've diluted the lysate down. And so as you can see, you have clear signal. The dark blue bars are the stimulated lysates, where they're stimulating this pathway, and the grayer bars are the unstimulated lysates. So we're comparing them. And as you can see, in the stimulated, you see clear phosphorylation of the receptor all the way down to like 10 nanograms of protein loaded into that sample. Again, we did the same thing for the phosphorylation of MEK, the phosphorylation of ERK, phosphorylation of P38, AKT, and J&K. All the assays that people routinely would run to understand these pathways. So then once we developed all these assays, then we wanted to really go further with the technology. And what we've done next is really looking at heterodimer interactions between these kinases. And that's what's illustrated here. And what we've done is you take the MEK assay. In the MEK assay, you have an antibody that's specific for the total MEK, and an antibody that's specific for the phosphorylation of a particular site. And that's one assay. In the other assay, we've looked at the phosphorylation of ERK. So we paired an antibody to phosphorylation site of ERK and total ERK. And then what we've done in a separate well is mix these reagents. So what we've done is we take an antibody to total MEK and an antibody to total ERK. And then what that allowed us to do, we know if they're pairing together and giving a signal that MEK and ERK must be interacting. So that's the data sets that's here. So we took two separate cell lines, we grew them in culture, we stimulated them either with EGF or we did not stimulate the EGF, and we looked for the detection of this heterodimer. And this is a standard dilution series of lysate, starting at 3 micrograms of lysate and diluting down, two separate cell lines. And as you can see, we have clear signal that we have heterodimer formation in both these cell lines, as well as even with stimulation of EGF, we can still see that and there's no change. So I think that's quite important to understand, that we can take this technology and not only detect an phosphorylation of a kinase, we can also detect interaction of kinases. So this is what we've done thus far. But lastly, what we've done is that we wanted to do was, can we look at all these pathways in a single plate with a set of samples? And that's what's illustrated here. We've taken these two same cell lines, stimulated them with EGF or not stimulated them with EGF, and then in every column we've put in a different assay. So you have a phosphorylation of AKT, JNK, P38, MEK, ERK, and the receptor. And what we've done, a different amount of lysates across the plate, and we've looked for the increase in signal. And so what I've illustrated over here is actually the signal. And then we graphically, by increasing the amount of color, shown how clearly you can see with stimulation, we have the phosphorylation of the receptor we showed with EGF. We have also classically the phosphorylation of MEK. That pathway is classically understood to be phosphorylated, with the phosphorylation of ERK, which is the same pathway. But we also have the phosphorylation of P38, which is a separate pathway. So you're able to see that as well. Don't see JNK lighting up, and we don't see a lot of AKT. So with single drug treatment with multiple cell lines under multiple lysate conditions, we're able to, in a single plate, look at 4 different pathways very easily with the technology, as well as look at interactions between kinases in the same assay platform. So that's pretty much the poster format. You're welcome.